you are interested in joining the Q&A afterwards. I think this is a really nice and great, it's a great opportunity for everyone who likes to discuss the work of others, but even their own work. And we never know when discussing with other people might also change the direction of our research. So I really encourage students and even postdocs or PIs to just jump into these discussions. Also, you can find different types of information regarding our talks on our website, which I will also drop in the chat box in a bit. And you can find their past talks. You can find the talks that were recorded. So if you want to watch them again, you can also sign up for our mailing list with the link that is also on our website in case you want to get our emails, advertising for the next talks, etc. So that's it. We might wait a couple more minutes for everyone to join. And as you probably know, the ones that attend our seminars regularly, we have had these polls in which we try to probe our audience a little bit to kind of get to know you, and you can also get to know us a bit. And today we were wondering, if you had to pick one, which do you consider the biggest challenge during your PhD? So I'm doing mine, starting mine right now, and all of them seem great choices. But yes, you can choose between time management, keeping up with the literature, writing, presenting your work, discussing your work, or others. I would like to take the opportunity to ask Dr. Michael what he felt while doing his PhD. What was the most difficult or biggest challenge?
Yeah, I would have to say, so I'm not sure really mine really shows up in the list, although maybe I voted for other. I guess dealing with failure, I think was actually the most challenging thing for me. Being able to adapt to the idea that most of what the experiments we try to do end up failing, and being able to move on from that and actually learn from those mistakes, or even not mistakes. Whenever you design an experiment and it fails, hopefully you learn something and move along. That can be a tough thing to learn.
That's an amazing input. I've been experiencing that myself. It's quite hard because I do relate with that, because often you kind of put your heart into a certain hypothesis and then it just doesn't work, or the experiment just failed, and it doesn't mean that you're not a good scientist. It means your hypothesis was wrong, and that's completely normal. You just have to find another one which might happen to be correct. But I agree, it's really hard. And I think the role of good mentors here, it's key and crucial, so that you have people that keep you on your track and kind of tell you what you just told everyone, which is normal to fail, which is not failing, it's learning throughout negative results, I would say.
Right.
Exactly. Okay, I think we are ready to start now. Most people might have already joined. So I'm going to present Dr. Michael. Dr. Michael is actually starting his lab quite soon, officially, at Yale's Department of Molecular and Cellular and Developmental Biology. He did his PhD, got his PhD from the University of Pennsylvania in cell and molecular biology, and afterwards joined the Brandeis University as a Ruth Al Kirstein, sorry if I pronounced it incorrectly, NRSA fellow in the lab of Dr. Piali Sengupta. There, he studied sensory neuromodulation by microbial metabolites using C. elegans. His work has included the discovery that neurotransmitters produced by gut bacteria can alter olfactory neuron function and feeding behavior. So, Dr. Michael, the stage is yours. Thank you so much.
Thank you, Rita. Okay, so I'll share my screen now and bring up my slides. And you can let me know if you see the presentation. Just might take a second. Can you see my slides now?
Right, all good.
Okay, great. Yeah, so thanks again, Rita. I gotta say, I'm really excited to be giving a talk in this seminar series. I think we were discussing a little bit ahead of time, Rita and I, about how it's been adapting to life amidst this pandemic, and I think that that series like these have been, sort of, if you can say a silver lining in this process. I think that having this great science and being able to be exposed to this kind of stuff, across the world has been really great over the course of this time. So as Rita said, I am a new assistant professor in the MCDB department at Yale, and I'll be officially starting my lab in January. And in my lab, what we work on is how bacteria can alter host behavior, and we study this using the nematode C. elegans. And so here's a picture of the posterior gut of C. elegans with some bacterial cells inside of it. And so I'll get to describing a little bit about how and why we use C. elegans in this process. But before I go into that, I just want to show a cartoon that maybe some of you have seen. It's fairly dated, and it's been used to analogize inequities in educational expectations. But I think it's actually a fairly apt example for neurobiologists if we sort of take these things a little bit more literally. And so one way we can sort of look at understanding the organisms that we study is to try to ask them to perform tasks which are relevant to the environment in which these animals originally evolved. And so we can sort of think of obvious examples of environmental parameters. Like, for example, of course, a fish can't climb a tree because it didn't evolve in an environment which contained trees. But there are many more sort of hidden environmental parameters, and the one that I want to focus on is the multitude of microbes which surround us both inside and out, and how these microbes might affect behavior of their hosts. So this is an area that's received a lot of attention lately, and so this is a field that has started to develop lots of evidence now that microbes not only can impact the physiology or the nutrition of their host, but they can also affect behavior. So there are examples of this both in vertebrates and invertebrates, but I would argue that up to this point, we don't yet have a great molecular handle on how these processes occur. So what are the ways that these microbes that, for example, inhabit the intestine of the organisms that we study, or even us, how do these microbes communicate with the nervous system in order to affect behaviors? So I'm particularly interested in understanding what are the microbial metabolites that are produced, and how can these metabolites interact with the nervous system of a host. And so before I talk about the system that I've been using, C. elegans, I just want to talk about my opinions about when I started this project, about how I would sort of imagine building up a system from scratch, and what kind of parameters we'd sort of like to have in that kind of a system. So in order to study how host and bacteria can together impact the behavior of an animal, you'd want to have, in my opinion, a number of different attributes to a particular system. So you'd like to be able to characterize the bacterial diversity and also manipulate the types of bacteria which are interacting with the system. So for example, you'd like to be able to sterilize and provide individual bacterial strains or mixtures of bacterial strains to your host. You'd also like to have access to to host genetics, so be able to take advantage of variation in host genetic background, but also manipulate the host genome in a really straightforward way. And of course, if you're going to study behavior, you'd like to have access to both high throughput and robust behavioral paradigms that you can use to try to study how these processes come together to affect responses of an animal. So I hope that by the end of this talk that I can convince you that C. elegans has many of these types of advantages. And that's really the reason why I've chosen to use this as my host organism to start my lab. But before I get into why C. elegans and what is the C. elegans natural microbiome, I just want to tell you a short story about how the worm lost its microbiome. And so this is a story that begins in 1951 with a mushroom and a worm. And this worm was originally isolated by Warwick Nicholas from a rotten mushroom in a class run by Ellen Stanland. And this single worm is essentially the founder worm for the strain called the Bristol or the N2 strain. And this is the ancestor of most of the worms that are studied in C. elegans labs throughout the world. So C. elegans feeds on bacteria. So originally Warwick was able to culture these worms on the bacterial contaminants from the rotting mushroom, from which he isolated the worm. Then together with using a protocol developed by Margaret Briggs, they developed a monoculture system using E. coli or a completely sterile system of an azenic culture in the absence of bacteria. And it was cultured for several more years before the strain was passed to Ellsworth Doherty, who together with Ada Hansen cultured this strain for the majority of the time in sterile conditions before passing the strain on to the man who made C. elegans famous, Sidney Brenner, in 1963. In Sidney Brenner's lab, this strain was cultured again on monocultures of one or two E. coli strains before it was eventually frozen by John Solston in 1969. So about 18 years of culture of this particular strain. And this monoculture of E. coli and C. elegans together has been a really useful system to study and understand the contributions of nutrition to processes such as metabolism or physiology or even behavior in worms. And the reason E. coli has been really useful as a model for nutrition in the worms is that E. coli is really efficiently digested by the worms. And one way you can visualize this digestion process is to label E. coli with a fluorescent protein. In this case, if we label E. coli with an mCherry expressing plasmid, and then feed these bacteria to the worm. So the worm actually sort of slurps up these bacterial cells and then passes these cells into the structure called the grinder. So this is at the posterior end of the pharynx here. And this structure actually crushes the bacterial cells. And what you see, if you look in the gut of these worms, is you see this diffuse red fluorescence. And that's really indicative of the majority of these cells being crushed. And so you can sort of think of these E. coli strains, which are typically used to culture C. elegans, as sort of tiny little bags of food for the worm. And so this system has been hugely useful throughout the years in understanding contributions of nutrition to various different processes which have been studied in the worm, such as metabolism, physiology, and behavior. But what if I told you that in the wild, E. coli is rarely found in association with C. elegans? Then I think maybe if we look at this time frame, then we sort of have to come up with a slightly different perspective on this culturing process. So it seems that if this worm strain was cultured for over 15 years in an environment which is very different than the environment in which it evolved, then it's basically undergone several thousand generations of replication before it was eventually frozen. So a lot of time to adapt to laboratory conditions. So I hope, and a number of other labs are taking this approach as well, to try to add back some of the natural microbiome to these worms, so we can actually study what effects these microbes might have on different processes such as behavior. So what is the natural environment for C. elegans, and what are its natural food sources? So we've really only come to understand this as a field more recently due to the work of several different labs, due to the sampling work of several different labs. And so these are a couple of pictures from a review from Henrik Scherlenberg and Marianne Feli. And their labs have played a large role in the sampling efforts for identifying what are the natural habitats of C. elegans. So you can find C. elegans in the wild in rotting plant material. So think rotting apples or compost. And you can find them in association with other invertebrates that sort of exploit these niches. So the reason why you find C. elegans in these places is that these are abundant sources of bacteria, which I mentioned to you are the primary food source for the worm. So bacteria are not only food for the worm, though. So bacteria can be both food, but also potential pathogen or parasite for the worm. And many different labs have studied the processes by which C. elegans defends against these diverse pathogenic threats. So pathogens in the worm can be bacteria, can be fungi, or even viruses. But I want to focus today on the role of commensal or mutualistic bacteria that we find in the intestine of the worm. So it turns out if you sample bacteria in the wild, unlike what you find typically in the lab, you find the intestine is colonized by a large number of bacterial cells. And so this is a fluorescent in situ hybridization showing bacterial cells in the intestine. And so you might think that the worm is so small, it can't possibly have a complex or specific microbiome. And so the work of several different labs has actually really established that the microbiome of the worm can be quite specific. So here's an example of that. So these are data showing principal component analysis, and so showing the bacterial diversity of bacteria that are found on different substrates. So for example, in blue here showing bacterial diversity on rotting fruit substrates, or in red here, compost. And what you'll notice is that these cluster together but separately. So the bacterial diversity is quite different on these two different substrates, which is maybe not surprising. But if you look at worms that are isolated from these individual substrates, you find that these tend to cluster together. So there's some host selection process which enriches for particular types of bacteria among these or from these substrate sources. So there's some role for the host in selecting the types of bacteria that inhabit the gut of the worm. So given that bacteria can be both food source and potential colonist of the intestine of the worm, I thought this really represented an interesting scenario in which the bacteria that inhabit the intestine of the worm could potentially impact the consumption of other bacteria or even their kin by impacting feeding decisions. And so given that this was an interesting possibility to me, I wanted to find a way to study this. So given that the worm doesn't have eyes, it uses primarily chemosensation to direct feeding decisions. And we can study chemosensation in the worm using these really robust behavioral assays. So here's an example of worms responding to an attractive odor in this case. And so what we do is we spot an odor to one side of an agar plate, and then we put a droplet of worms onto this plate and give them time to navigate. And hopefully this video plays here. And so we give them time to navigate in response to the odor. And you can see that the worms, this video is sped up, but you can see that worms relatively quickly and successfully make their way to the odor. So we can do these kinds of assays in a high throughput way where we simply count the number of worms at the odor side relative to the control. Or we can track the trajectories of these worms and look at postural data, such as in this case, showing you pirouettes, which is a type of turn that the worm engages in. We can even look at kinematic parameters, like in this case, showing you angular velocity, and try to understand how these parameters change in response to the heading changes of the worms. So for example, when the worms are pointed towards this attractive cue, there's a reduction in the angular velocity, which you expect for an attractive cue. So these behavioral decisions are driven by chemosensory neurons, which are located in the head of the worm. And so these neurons have their cell bodies really close to that grinder organ that I showed you before. And then they send an axonal process to a neuropil called the nerve ring, which is the synaptic region for these sensory neurons. And then they also send a long dendrite out to the tip of the nose, which is where the sensory endings are located. So I just want to give you a sense of the compactness and the scale of these sensory endings. And so I want to highlight some of the work of David Doroka, one of my colleagues. And this is serial electron microscopy showing the sensory endings at the tip of the nose here. And together with Denny Nicastro's lab in the Sigupta lab, David was able to reconstruct the sensory system on the anterior end of the nose And what you can see here are all the sensory endings of the chemosensory system of the worm. So these are the neurons that are detecting the chemicals that we assay for behavior. And so if you were a bacterium, so looking from head on from the tip of the nose, this is approximately what you would see before you were consumed. And you can see here, this is the entrance to the pharynx of the worm. So these chemosensory neurons detect both water-soluble and volatile chemicals. And so these volatile chemicals are diverse, but interestingly, many of these both attractant and repellent chemicals to which the worm responds are produced by bacteria that it may consume. So I reasoned that by testing for chemosensory behavioral changes, I might be able to use chemosensory behaviors as a proxy for effects on feeding decisions in the worm. So based on this, I wanted to ask the main question, I think that'll be the focus of this talk, is how can the worm's natural microbes, or do the worm's natural microbes alter the process of chemosensation, and does that affect feeding behavior in the worm? So to test this, I worked together with a fantastic technician, Henry Chow, and together we screened a large number of non-pathogenic bacterial strains and a large number of odorant cues, both attractive and repellent odors. And the way we set up this experiment was we allowed animals to feed for a single generation on a number of different non-pathogenic strains. And then we did this in a high throughput way. So we screened for endpoint chemotaxis on these agar plates. And we generated a chemotaxis index that I mentioned before, but this chemotaxis index is really calculated just based on the difference of the number of worms on the control versus the odor spot divided by the total. And just to give you a sense of the direction, so positive numbers for this index indicate attraction and negative numbers indicate repulsion. So I won't show you all of the data because we tested a large number of odorants, but I'll just point out a couple of typical responses and point out something interesting that we found. So we really didn't see across these panels of attractive odors, we didn't see much of an effect, but I'll point out this effect that we saw that was relatively specific to this 8-carbon alcohol called Octanol. So worms are normally strongly repelled by Octanol, but you can see in blue here is when we feed worms this Providencia strain called JUB39, worms, this reduction, this aversion to this alcohol is strongly reduced. So this bacterial strain was originally isolated from C. elegans by Marianne Feli's group, and it was carefully characterized by Buck Samuel's lab as a so-called beneficial bacterium. So it's beneficial because it increases the growth rate of worms, but it also protects against pathogens as well. And so I wondered whether, given that this is a nutritious food source, we wondered whether this was simply a nutritive effect or whether this required some other aspect of metabolism of the worm. I'm sorry, of the bacteria in this case. So we tested whether killing the bacteria had any effect on this modulation. Just to reorient you here, now I'm showing these chemotaxis index relative to the control condition, just so you can more easily visualize the effects of the treatment here. So pre-killing E. coli had no effect on the chemosensory behavior in this case. But if we pre-kill Providencia, this completely blocked the behavioral modulation in response to Octanol. So it turns out that the bacteria need to be alive to reduce Octanol avoidance in this case. So given that the bacteria need to be alive, I wanted to see whether they could colonize the intestine of the worm. And so we used a similar technique that I described to you before, where we fed the worms this M-cherry expressing bacteria, either E. coli or Providencia. And then we just looked in the intestine of the worm to see if we could find intact cells. So, and what you can see is there's a large number of intact bacterial cells in the Providencia grown or the JU39 grown worms. And we think that these bacteria are alive because if we bleach the worms externally and then grind them up, we can isolate large numbers of bacterial cells after grinding them up. But what we were really interested in was identifying whether this colonization process had any impact on the behavior. And so to test this, instead of grinding up the worms, what we did was we allowed the worms to be colonized by Providencia, and then we subjected them to a chemosensory gradient of Octanol. And if we then pick the worms off of this plate of Octanol and compare the animals on the control side versus the Octanol side of the plate, what we saw is that the animals on the Octanol side of the plate had a larger number of bacterial cells in their gut. So this suggests that the colonization process is correlated with the behavioral change in these worms. Okay, so we have this bacterium which is inhabiting the intestine of the worm, and then we have a change in behavior. So the question is, how can we get from this process of colonization to a mechanism of this behavioral change? And so I'll just walk you through in sort of 3 parts how we actually went about establishing this, and the 3 different sort of techniques that we used. So first, starting with worm neurogenetics to identify the modulatory pathways that are involved in this process, and then using metabolomics to identify metabolites, and then using bacterial genetics to disrupt the production of these metabolites. So starting with worm neurogenetics, so I think that a great starting point to try to understand how an individual behavior can be modulated is to start with the neuronal connectome of the worm. And so C. elegans has a complete connectome, and one way to visualize this connectome is to look in a so-called information flow diagram, which hopefully this loads. This next slide, which is taking a long time. Sorry, this is seems to be delayed in transitioning to the next slide. So yeah, okay, here, great. So so this is a way to look at the connectome of the worm that incorporates the information flow essentially from sensory to motor output. So in this information flow diagram, you can see the sensory neurons are colored in red, the interneurons are colored in blue, and motor neurons are colored in green, and information essentially flows from sensory to motor to drive behavior. And what you'll notice is that there are 2 different clusters of these green cells, these motor neurons here, and these actually correspond to 2 of the main locomotory systems in the worm. Those motor neurons that control head turning and those that control forward and reverse locomotion. So I find it's a useful way so if you have a change in a long range chemotaxis behavior, one way to try to understand how this process occurs is to look at these acute locomotory changes that might occur. And if we look at the olfactory subnetwork, so these are just the olfactory neurons, the olfactory sensory neurons here, you can see that these neurons also engage and communicate with both of these locomotory outputs. So how can we look at acute locomotion? So one way to look at this is to do a so-called smell on a stick assay. And in this assay, what we do is we take an aversive odor and we take a hair and we dip it into this aversive odor and present it in front of the nose of the worm. And then we just measure the amount of time it takes for the worm to back up in response to that odor. And this is sort of a measure of the strength of the avoidance response of worms. So for those of you who've done behavioral analysis before, you know that behavior is quite variable. But luckily in C. elegans, we can measure hundreds of animals and develop a distribution of these behavioral responses, which gives us some confidence in the effect of a particular manipulation. So in this case, what you can see is the response times for worms that are fed Providencia are shifted to the right. So it takes them longer on average to respond to Octanol. And this is consistent with the on-plate chemotaxis results. So if we take these distribution data and we flip them on their side and we look at them on the log scale, you can see what the relative latency is comparing the E. coli control to the Providencia grown strains. And this corresponds to about a 2 to 3 second delay in response to Octanol. And just to give you a reference, that 2 to 3 second delay is similar in magnitude to what would occur if you ablated the main sensory neuron called ASH in this process. So I think it's a meaningful change in the behavioral response. So we have this bacteria that is altering the reversal motor circuit to regulate octanol-driven behavior. So how is it doing this? And so, so one way to look at how it's doing this is to look at the pathways that we know already that have already been established that we know modulate octanol behavior. And so what I'm showing you here is, if it ever loads, is in vivo calcium imaging in the ASH sensory neuron. And this sensory neuron directly responds to octanol, but it's also modulated by octanol as well, and modulated by a number of different monoamine pathways. So here's the response in that neuron. So you can see the GCAMP response in response to a pulse of octanol here. So there are a number of different modulatory pathways that are involved in regulating responses to this chemical. And I'll just focus on two of these monoamines, octopamine and tyramine. And so these monoamines, you can sort of think of these as analogous to the adrenergic system in vertebrates. And these have been well studied in C. elegans and other invertebrates as neuromodulators. And one way that we can look at the function of these two monoamine pathways is to disrupt their function by mutating the enzymes that are required for their production. So tyramine is produced and octopamine are produced in a linear biosynthetic pathway from the precursor chemical tyrosine, so this amino acid. And so the enzyme TDC1, which is a tyrosine decarboxylase, generates tyramine. And then TBH1 further modifies this to generate octopamine. So for example, if we look in TBH1 mutants, those would be predicted to specifically affect octopamine production, but TDC1 mutants should affect production of both tyramine and octopamine. So we can see what the relative contribution of these two monoamines might be in this behavior. So again, if we look at this these reversal latency assays, these SOS assays, so what we can do is we can test first whether octopamine is required for these behaviors. So if we look in TBH1 mutants, we see that the modulation is essentially gone. So it's largely reduced here in this case. And so it appears that octopamine is necessary for this behavioral modulation by Providencia. And this is where things get interesting. So if you look in TDC1 mutants, and I'll remind you that TDC1 should disrupt production of both octopamine and tyramine. Essentially, TDC1 mutants look wild type for this behavior. So one possibility is that in TBH1 mutants, there's an accumulation of tyramine that results from a blockage of the biosynthesis of octopamine. And one way to test for that accumulation of tyramine is to look in double mutants. But if we look in double mutants, the behavior is also blocked. So it really looks like it's the loss of octopamine and probably not the accumulation of host-derived tyramine that's responsible for this behavioral modulation. So we have octopamine in some way inhibiting the reversal motor circuit. But then where is TA coming from? Where is tyramine coming from? Because we have no other known biosynthetic pathway to generate octopamine in the worm. So we thought that maybe the most reasonable place that the tyramine could be coming from was directly from the bacteria in this case. So to deter, and that might be bypassing the requirement for the host enzyme in this case in the biosynthesis of octopamine. So we collaborated with Bennett Fox and Frank Schroeder's lab to use targeted metabolomics to identify whether tyramine levels were altered by culturing on Providencia. And to do this, we used high-resolution HPLC-MS, and we looked for tyramine and tyramine derivatives in worms that are fed E. coli or Providencia. So in the control condition here, you can see we can detect tyramine both in E. coli-fed worms and Providencia-fed worms. And consistent with previously published data in TDC1 mutant worms, which are fed E. coli, we can no longer detect tyramine or tyramine derivatives. But when we feed these TDC1 mutant worms Providencia, the levels of tyramine are largely recovered in these worms. So this suggests that the Providencia can complement the host production of tyramine to some extent. And so we think that the tyramine is coming from a decarboxylation of tyrosine, because if we label the media with a heavy tyrosine label, we then see accumulation of heavy tyramine in the worm. So it looks like it's the result of a decarboxylation reaction, similar to what happens normally in the worm. So we have tyramine coming from the bacteria, driving these behavioral changes indirectly. So I wanted to understand how this bacterium could be generating tyramine. And using bacterial genetics, we tried to identify how these metabolites are produced in these bacteria. So first, I wanted to know whether this effect generalized across Providencia strains, or if this was sort of an esoteric or peculiar attribute of this individual Providencia strain. So I needed more Providencia strains, and so I went to my compost and isolated nematodes and their associated bacteria from the compost, and then found an additional Providencia strain, which is distantly related to the JUE39 strain. So in this case, it's a Providencia redgary strain. So when we test this strain in Octanol avoidance assays, you can see that the behavior is essentially the same as the JU39. So I thought this was good enough evidence that this trait probably generalized to a large extent across Providencia, and that it was worth looking in the genomes of these strains to identify enzymes that might be responsible for this. So how do we know where to look, and what should these enzymes look like? So luckily, there's been a lot of work done in the food science literature in understanding how monoamines like tyramine can be produced by bacteria. And so monoamines like tyramine are known spoilage agents in foods. And we've actually known for nearly 100 years that bacteria had the biosynthetic capacity to produce monoamines like tyramine. And this has been well studied in gram-positive lactobacillus and enterococcus strains. And there's a conserved operon that is responsible for tyrosine production in these strains. So this operon contains an enzyme called tyrosine decarboxylase. And this enzyme is homologous to the eukaryotic enzyme that I talked about before, this TDC1 enzyme. So this is an ancient enzyme family. And then this enzyme is always found in, or nearly always found in an operon containing a permease. So this is a permease which exchanges tyrosine for tyramine. So this is a way that the cell can take in tyrosine and export tyramine. So we looked for this type of operon signature for enzymes that looked like a decarboxylase enzyme. And so we sequenced the genomes of those two Providencia strains, and then in their genomes, we found two different types of decarboxylases. So both of these decarboxylases are found in some cases in operons with a permease, so a predicted tyrosine permease. And so one type, which we'll call the enterococcus type, is highly homologous to the gram-positive tyrosine decarboxylase enzyme. And so we think that this gene was probably acquired by horizontal gene transfer in the Providencia clade. There's another enzyme which is more distantly related that's sparsely found in Providencia, but is almost uniformly found in the closely related Morganella. And this enzyme, which is a decarboxylase enzyme, is often found in an operon with a type 1, so a different type of tyrosine transferase. Or putative tyrosine transferase in this case. So given that both of these enzymes had this characteristic operon structure, we wanted to identify whether these enzymes were responsible for the behavioral modulation that we see in C. elegans. So we made knockouts of these two genes in the JU39 strain, and we tested the effects of these knockouts on behavior. So again, what I'm showing you is reversal latency. And so this is the 2 to 3 second delay that you see when you feed animals Providencia in response to Octanol. And if we look at either of the single mutants for these enzymes, the effect is about halved. But if we look in double mutants where both enzymes are knocked out, the effect is essentially gone in this case. So it looks like the activity of one or both of these enzymes is necessary for this behavioral modulation of these worms. Importantly, in these bacterial knockouts, we can no longer detect any tyramine. So it looks like these two enzymes are required for tyramine production by the bacteria. So we identified enzymes, tyrosine decarboxylase enzymes that are necessary to convert tyrosine to tyramine, and then that's what's responsible for driving these behavioral changes in the worm. So given that it looks like tyramine is the chemical that's responsible for these effects, we wondered whether we could just bypass the requirement for the bacteria altogether and just add tyramine to plates. And so to do that, we added tyramine to E. coli plates and tested the octanol avoidance behaviors under these conditions. And we saw a delay in octanol responses that were similar to what has been previously shown, what we previously showed with Providencia. Interestingly, if we add tyramine to cultures of wild-type Providencia, there's no effect on octanol avoidance responses. And these results are actually normalized to the no tyramine control, just to show you the effect of the drug treatment or the amino acid or the monoamine treatment. When we add tyramine to TDC knockout bacteria, we can recover the behavioral modulation. So together, this suggests that tyramine is both necessary and sufficient for octanol modulation under these conditions. So I showed you data implicating octopamine in this behavioral response. So what are the receptors and where are these receptors acting? And so going back to worm genetics, we wanted to look at the ASH sensory neurons themselves. So I told you before that ASH is the primary sensory neuron for detection of octanol, but it's also the site, one of the sites of neuromodulation by these monoamines. So ASH actually expresses two different octopamine receptors and a single tyramine receptor. And in data that I won't show you, we found that the octopamine receptor SER3 or the tyramine receptor TYRA2 were not necessary for this behavioral modulation, but that OCTR1 was. So again, this is reversal latency data showing you here, and this is what the data looks like in an OCTR1 mutant. And so you can see that there's no longer any delay in octanol avoidance behavior responses in OCTR1 mutants. We can rescue this receptor by re-expressing this receptor just in the ASH sensory neuron. So it looks like it's acting in ASH directly to regulate these reversal responses here. So it looks like octopamine is signaling through the OCTR1 G protein coupled receptor to regulate reversals in response to octanol. But in the beginning, I mentioned to you that I was really interested in identifying cases in which feeding decisions could be altered by colonization of bacteria. So luckily, we know from work from Elizabeth Glader's lab that Providencia produces a number of different odorants, and some of these can be actually aversive to C. elegans at high concentrations. So I wanted to test whether colonization of the intestine by Providencia impacted feeding decisions on these actual bacteria. And to do that, we used a sort of a simple food choice assay. In this case, we again fed worms these different bacterial strains, so either E. coli or the different Providencia strains. And then we tested a short-range food choice assay where animals were allowed to choose either the control E. coli spot or a Providencia spot. And I gave them 15 minutes to execute this behavior. So what we found is relative to the E. coli fed worms, we find that Providencia, animals that had been fed Providencia for a single generation tended to prefer Providencia spot. But this only occurred in animals that were fed Providencia that can produce tyramine. So it seems like tyramine is required for this behavior as well. We also found that this behavior required this octopaminergic signaling pathway. So it requires the OCTR1 receptor and also requires host production of octopamine. So it looks like both octanol modulation and food preference changes act via essentially the same biological circuit to regulate these behavioral changes. So everything that I've shown you involves the effect of octopamine in the worm. But of course, tyramine is a neuromodulator and a neurotransmitter in its own right. And so I wondered whether tyramine could affect behaviors directly in the worm. So to test this, we wanted to identify any cases in which bacterially derived tyramine might be able to compensate for the loss of host production of tyramine. So we used a well-characterized behavioral paradigm in which tyramine regulates egg-laying behavior in the worm. So tyramine inhibits egg-laying behavior by interacting with the HSN neuron. And this is established. This has been established by work from Mark Alkema and Kevin Collins and Michael Cole's lab. So, so one way to look at the effects of tyramine on egg laying is to look in the uterus of the worms and look at the average average age of the eggs in the uterus. And so in this case, I'm quantifying the proportion of eggs that are over the 4-cell stage, and we chose this stage based on previously published data. So if you compare wild type worms to TDC1 mutant worms in this case, you can see that the eggs are on average younger in TDC1 mutants. And this is consistent with an effect of tyramine on inhibiting egg laying in this case. If we feed TDC1 mutants wild type Providencia, the egg laying age, or the average age of the eggs is recovered, but this only occurs in wild type Providencia, so not in the tyramine production knockout bacteria. So together, what I've shown you here is that these bacteria can produce a metabolite using their own biosynthetic machinery that can either directly affect a behavior. So in this case, I showed you that tyramine produced by Providencia can impact egg-laying behavior directly, but that these metabolites that are produced by bacteria can also participate in host metabolism. So using the host biosynthetic machinery, tyramine is likely converted to octopamine, and then this can affect different behavioral decisions. So a point that I want to emphasize based on this is that that in my opinion, it's really important to consider the effects of joint metabolism. So the cooperative biosynthetic production of neuroactive chemicals by both bacteria and the host might lead us to understand a little bit better how these two processes can come together to affect behavior. And I think when we look at the biosynthetic network, or the metabolomic network of C. elegans, which are fed Providencia, I think that this joint host and microbe metabolism process really stands out, and I think it's actually pretty striking. So this is some analysis that was done by Bennett Fox in collaboration with Max Help, who developed a software called MetaboSeq, and this MetaboSeq software uses the MS2 spectra from HPLC-MS, and importantly, what it does is this software clusters similar chemicals just based on their MS2 spectra. So you really have you really don't need to have any prior knowledge of the chemical structure in order to cluster chemicals in using this software. So so what we found, which I think is pretty striking when looking at this metabolomic network data, is that if we look at a single cluster that really stood out from all of the other thousands of products which we were able to detect in these metabolomics analysis, and we found a large number of structurally related chemicals which were altered in a Providencia and tyrosine decarboxylase dependent way. So so what are these chemicals, and what are the patterns of their production look like? So interestingly, if we look at the production of these chemicals that are located in this cluster, we find that all of them require, under E. coli conditions, E. coli grown control conditions, all of them require the host TDC activity in order to be produced. And in large part, most of these chemicals are actually recovered when we culture these animals on wild type Providencia, but not so in bacteria that can't produce tyramine. So if we then go and look at the predicted structures for all of these 16 chemicals, you find that every one of these chemicals has a predicted tyramine moiety. And we confirmed in a large number of these cases that there's using the heavy tyrosine labeling, that these are likely tyramine in these chemicals. So maybe this isn't surprising based on the sort of specificity of the effects of the tyramine knockouts that I showed you before. But this was actually a shocking result to me because I expected there to be thousands of chemicals which were changed in these knockouts. But this really argues that I think to me that this kind of approach can be really useful for identifying specific metabolic pathways that can be manipulated or disrupted by changing biosynthesis in both the host and the bacterium in these cases. So I just want to wrap up real quickly, but just to go over an overview of the directions that I think that I plan on taking this work in my own lab. And what are the sort of the types of things that I think we can discover using this system. So I think we've really only scratched the surface of by doing this analysis on the tyramine pathways. So we really are sort of poised in this elegans field to look at natural variation in host genetics and try to understand how natural variation both impacts the assembly of the microbiome in the worm, but also how this host genetic background can impact behavioral changes in response to bacteria. And so what we'd like to do is incorporate both differences in natural variation in host genetics, but also expand the bacterial diversity that we've looked at. So to use combinations of microbes or larger numbers of bacteria to assemble more complex microbial communities. And all in my lab, I plan to use these tools to try to understand how these processes can impact behavior and how these processes can be interdependent. So whether behavior actually drives the assembly of a microbiome and how host genetics plays a role in that process. And I think in the process of studying this, I think we can learn about new ways of neuromodulation. We can possibly identify new immune pathways that drive microbial assembly of the microbiome in the worms. And as I showed you, we can identify cases in which metabolites can impact the behavior of worms. So with that, I'll just go to my concluding slide. And I just want to thank the people that helped me in this process. So I pointed out Henry Chow. Henry was hugely useful, hugely useful. So Henry was a great help in the beginning of this project, and he helped a lot with this initial screen. I want to thank the members of the Sengupta lab, Piali, and the rest of the members, and also former members of the Sengupta lab who have been really helpful colleagues, and I'm going to miss them as I move on to my own lab. So I've worked with a number of great collaborators who enabled me to do this work, and so I do want to acknowledge them. And I also want to acknowledge that that a lot of this work is on the shoulders of a lot of great chemosensory and genetic work and sort of wild sampling work that have been done by a large number of labs in the C. elegans community. And I've been really happy with the C. elegans community in this aspect of how generous and in sharing of reagents and also in sharing of unpublished data that can be. I don't want to forget to mention that my department, MCDB at Yale, is hiring. So those of you that are on the job market this year, so we have a great department, and if you're on the job market, please apply. And so I think the deadline for the application is November 15th. So come join us. And with that, I'll end screen sharing.
Thank you so much for the very interesting and nice talk. Really, really nice.
Thanks,
thank you. It's quite nice to hear about something that we also explore in the lab, and it's super interesting. I had no, well, I read the paper, but it's easier with you going through it. So we have some questions. Maybe we can start going through them. So Zita Santos asked, maybe I missed it, but have you checked the levels of tyramine in wild-type animals versus bacteria colonized ones, not TDC mutants? Also, have you checked what happens to tyrosine?
Yeah, so tyrosine levels we haven't actually directly looked at, although that should be in the data set, so we could look at that. So interestingly, so it's a lot easier for us to detect this succinylated tyramine, so it's abundant at much higher levels. And so the data that I showed you about the abundance of tyramine was actually the succinylated tyramine, so it's a byproduct of tyramine. And so we actually see similar levels of that byproduct in wild type worms that are fed Providencia. So it's possible that they're processing enough of it to convert into octopamine, so that we're only really detecting sort of steady state levels of tyramine in that case. And it's really only in the TDC mutant worms where we see this dramatic effect of increased tyramine. That's because there's essentially none in the wild type, in the knockout worms in this case.
Okay, great. So another question from Haluk Lassin. What other animals do these bacteria colonize? Other invertebrates? Do you know if they change behavior of other animals? I guess it's referring to Providencia.
Yeah, so I'd love to know that. So Providencia is actually so it's a fairly common pathogen of insects. So it's a pathogen for Drosophilids, and so yeah, so I'd be really interested to look at whether so it's interesting though it doesn't appear to be an intestinal pathogen for Drosophilids. It tends to circulate in the hemolymph, and that's I don't know about these particular bacterials. I mean, I isolated these from, well, not these. I isolated one of these strains from nematodes. I don't know whether these are pathogenic towards insects or not, but there are pathogenic Providencia strains that affect insects. So Providencia is also, so it occasionally can be pathogenic to humans in some certain cases. It's mostly nosocomial infections. We don't know any other cases in which this bacteria in the intestine, alters behaviors as of yet. But yeah, this is an area I'd love to look into.
Cool. Okay, another question. Maybe I missed. I know this was already answered. Sorry. Have you considered the hypothesis of TA could drive signaling directly to TA receptors in some key sensory neurons by Fernando Calahorro?
Yeah, so that's certainly possible. I think that so one issue is we really only looked at one type of behavior in terms of chemosensory behaviors. So we've only really looked at Octanol avoidance. I mean, we had some controls with other odorants. We think this effect is relatively specific to Octanol avoidance, but Tyramine drives lots of different behavioral changes. So for example, so Tyramine acts as a fast-acting neurotransmitter in driving suppression of head reversals in response to nose touch, and so we haven't. So I don't think that it's as likely that Tyramine would be affecting those kinds of behaviors, because what I think is probably happening is that you have circulating levels of Tyramine in the worm. Sort of in the pseudocoelom of the worm, and that this can sort of act as a sort of a neuromodulator. Whether it's taken up into neurons and packaged into synaptic vesicles, we don't know. So that's really an interesting question. Is how is it taken up into neurons, and how is it converted into octopamine? Are both of these packed into synaptic vesicles? And so we're working on a couple of different labeling techniques that we can, or reporter techniques, we can actually try to watch that process happen. But it seems like in some cases, Tyramine is just able to diffuse through cell membranes. So it's also possible that it's just passively diffusing through the tissues as well.
Cool, interesting. Okay, final question by Eamon Fitzgerald. Great talk, thank you. Do you know if these interactions or interactions like this are under positive evolutionary selection?
Yeah, I wish I knew that. So this is a really tough thing to test. So I guess I don't want to overstate the interaction of this bacteria with the worm. So I mentioned to you in the beginning of the introduction that worms have a really diverse microbiome, and so I think that this is not so much a symbiosis effect between Providencia, but more of an opportunistic effect. And so I can imagine situations where this would be beneficial for the worm, but in terms of directly demonstrating it, yeah, we don't have that data that would indicate that's the case. So there are some cases where Providencia has what looks like more of a symbiotic relationship with nematodes. And so one example is in heterorhabditis. These insect pathogenic nematodes, they're very commonly found co-colonized by Providencia strains as well. So that's sort of an interesting thing, aspect of Providencia, sort of how is it establishing more stable symbiotic interactions.
Cool, really interesting. Okay, I think those were all the questions for now. Now we will move on to the Q&A. Start moving there. Thank you, Dr. Mark, clear. That was really nice. And I would like to tell everyone that you can join us next week now at our regular time. Sorry for the messing up the schedule because of the hours changing, but I'm happy that everyone ended up joining. And next week we'll be hearing about Dr. Yoav Livne, work, whom has recently joined the Weizmann Institute to study how the brain, particularly the insular cortex, perceives and integrates different bodily signals such as heart rate, temperature, thirst, and the relevance of this in health and disease. So thank you once again, Dr. Michael, and I will see everyone in the Q&A. Bye-bye.